So the topic I chose is co-immunoprecipitation and immunoblotting. So a bit of background on both of these topics. Co-immunoprecipitation, or Co-IP, is a technique used to identify physiologically relevant protein-to-protein interactions by using target protein-specific antibodies to indirectly capture proteins that are bound to a specific target protein. These protein complexes can be then observed and utilized to identify many factors, such as identifying new binding partners, binding affinities, the kinetics of binding, and the function of the target protein. Co-IP is then followed with immunoblotting to further observe these interactions. Immunoblotting, or more well known as the Western blot, is often used in research to separate and identify proteins, and in this technique, a mixture of proteins is separated based on their molecular weight and as well as by type through another procedure known as gel electrophoresis. Application and principle. So co-IP involves allowing sample, wash, and other solutions to pass through a column that is packed with porous resin, typically beaded agarose, onto which a target-specific antibody has been immobilized. A small amount of resin in a microcentrifuge tube is used, and incubation steps are performed in a batch-wise manner. For each step, solution is added to the beads, which are then mixed and incubated together where the beads are suspended in the solution. At the end of each incubation step, the beads are pelleted to the bottom of the tube by centrifugation, so the solution can be removed using a pipette. The goal of immunoprecipitation is to isolate just enough protein to be able to measure using the immunoblotting method that we have discussed. Usually, treated and untreated samples are compared to assess the relative amount of the protein of interest. And so a bit more on the immunoblotting is it is performed by preparing a sample from cells, separation of proteins by size on the SDS, or sodium thiodecyl sulfate, polyacrylamide gel by electrophoresis, immobilization of separated proteins in a nitrocellulose or polyvinylidene fluoride, or PVDF, membrane, blocking of nonspecific proteins on a membrane, probing of target proteins with specific primary antibodies, incubation with secondary antibody conjugated with labeled chemiluminescent or fluorescent molecule, and detection of signal that reflects antigen or antibody binding, and densitometry analysis of protein bands of interest using a software. So this figure is a figure from an article called Co-immunoprecipitation of the mouse MX1 protein with the influenza A virus nucleoprotein. So this article investigates the interaction between the antiviral myxovirus-resistant protein, or MX1, and one of its viral targets, the influenza A virus nucleoprotein, or NP. The experiment performed for the study includes injecting mammalian cells with the influenza A virus, and the authors also include that cells already infected can also be utilized to begin the study. After a cell lysis, the viral NP protein is pulled down with a specific antibody, and the resulting immune complexes are precipitated with the protein G beads, and this process is the co-immunoprecipitation process. Once this is successfully performed, the co-IP with the antiviral MX1 protein are subsequently revealed by the Western blotting, or immunoblotting. Co-IP and immunoblotting can be used to study interactions between MX1 and the influenza NP protein isolated from infected cells or from virons. For this application, the protocol was slightly adapted by combining lysates of MX1 expressing cells with lysates containing the viral NP protein before starting the co-immunoprecipitation protocol. The figure over here on the left shows the co-IP of MX1 with NP isolated from transfected cells, infected cells, or virons. And so for some strengths of the study are that they show that this co-immunoprecipitation protocol can be successfully used to study the interaction between an antiviral protein and its viral target, shown here in the figure. And some drawbacks and limitation of the experiment were the inadvertent pull down of contaminating proteins caused by nonspecific binding of proteins to the protein G beads or antibodies. The authors included this is very important to include control settings to exclude false positive results and any contamination in the results. And so one question I do have is, aside from the Western blotting, what other techniques can be used to further observe interactions following the co-immunoprecipitation analysis?